Okay, so it's 12. Marta, do you want to do introductions?
Yes, sure. I'm ready.
Go for it.
Okay. Then we are very excited to continue the SRF webinar series. The goals of the series are: 1, getting you closer to the science; 2, making you aware of the research that is being done and the opportunities to participate; and 3, empower your communications with clinicians. We also want to remind you that our next webinar in the series is with Dr. Stefan Sanders on SynGAP1 in the Developing Human Cortex tomorrow, Friday, November 6th at 9 a.m. Pacific. The talk today is how and why to use human cells models to study SynGAP1 by Dr. Marcelo Pablo Coba. Dr. Coba is an associate professor of psychiatry and behavioral science at the University of Southern California, Keck School of Medicine. Dr. Coba's impressive educational background includes a doctoral of pharmacy and postdoctoral work in neuroscience. His education sets him apart and aligns well with SynGAP patients' needs. Dr. Coba has a long-standing interest in the role of proteins like SynGAP1 that are important in the synaptic function, synaptic plasticity, learning, and memory. He wants to help us to understand the molecular and cellular changes that occur in our SynGAPians. Professor Coba proposed a plan to address these essential and complicated questions. The proposed work includes groundbreaking new ways to describe the effects of non-pathogenic SynGAP1 variants in the neuronal cells, and critical to find molecular targets for potential medications. Dr. Coba has a close working relationship with SRF and has been generous sharing his ideas and listening to parents. For several years, Dr. Coba has been working on SynGAP1 encephalopathy. Now, he's asking the SynGAP Research Fund to support his work. He's optimistic this will eventually lead to better outcomes in translational research. We as a group feel privileged and lucky to have Dr. Coba working with us, and we are thrilled to support his work. After this brief introduction, I want to let you know a recorded version of this webinar will be available on the SRF website. By the end of the presentation, you will have opportunity to get your questions answered. We love to hear from you. Please write your questions in the chat. For those of you just joined us, welcome. And our talk today is how and why to use human cellular models to study SynGAP1 by Dr. Marcelo Pablo Coba. Okay, Marcelo.
Thank you, Marta, and thank you all the guys at SRF for the invitation. Yeah, I will just will be some briefly just some brief things about how to use it, or what are we going to do, or what we are doing with human models, and things that at least to try to make clear what we can get, what we are trying to do, but hopefully it's not in a very complicated way. So we're going to briefly, this is the outline what I'm going to discuss. Briefly, I'm going to say, well, how has been my path with SynGAP and my rediscovery of SynGAP in the last years. And I will give my introduction of SynGAP1. I'm sure that you have an introduction of SynGAP1 from every speaker, and you know really a lot. And I think that this is going to be a bit different. And then we're going to describe a bit about the molecular composition of the synapses, the kind of synapses that we have in these human models, how we can use them, approaches, and a couple of examples on mutations at SynGAP and the kind of phenotypes that we can see in these cellular models. So when I started with SynGAP, I bumped into SynGAP. I'm originally from Argentina, and I moved to England, to Cambridge, UK, at this place, which is the Wellcome Trust Sanger Institute, where guys play a big part in decoding the human genome in the early 2000s. And I moved to work with, I don't know if you can read it, with at the laboratory of Seth Grant, which has an interest in synaptic signaling, and he started to do different models of synaptic dysfunction during mutations in different components of the synapse. And in 2002, he published the first animal model of SYNGAP. It was a haplosufficient model. So it was a mutation in SYNGAP that wiped out around 50% of the total protein levels, which is many of patients have that particular phenotype. And as I said, at that time, there were very few people working on SYNGAP. Probably was Rick Hugonir, later is Gavin coming from that group, Mary Kennedy and Seth, who got the first animal model. And it was, if you want, a low-key publication. It's shown on Neuroscience. It's not one of the top journals. So just to have an idea of what was the interest and the field on SYNGAP. Not many people working on that particular gene protein, and also you don't get the best publications on studying that molecule. But my interest was in synaptic function. Really, I didn't move there just to work on SYNGAP. I was just trying to address just general mechanisms of how the synapse work, in particular, the postsynaptic side. So you have a presynaptic side, and at the other side of the synapse, you have the postsynaptic side. So mostly working on that side of the synapse, trying to address mechanisms of learning, memory, what we define as synaptic plasticity, very basic science questions. And SYNGAP was one of these molecules involved. So we've been around with that more. When I moved there, SYNGAP was one of these molecules. The group was trying to knock down or just get mutation to as many components of the synapse as possible. So SYNGAP was really another one. We were used to compare results with other models. They were always around with the SYNGAP bounce model. But I always remember one day, and it was in 2009, that I bumped into that publication and really struck me, because at that point we were doing really what I consider basic neuroscience, addressing mechanisms, really intricate mechanisms. If you have one receptor being more or less receptor of the membrane, what is happening with the electrical activity of the synapse, this kind of thing with each particular mutant model. But then we got this publication, it was in 2009. It was the first report of patients with mutation in SYNGAP. And I remember that day sitting at the office, and it was to me like we were working with an animal model, and I can see that some of these patients that are reported in that paper had really a similar mutation that it was in the animal model that we were studying. That is the haplosufficient model. And that was, to me, the first at least realizing that what we were doing, it has this connection to real people, not just we were doing basic mechanism in synapses. And a main thing that happened, you can see that the first paper came in 2009, and then in 2002, at least the animal model, Rick and Mary Kennedy published on SYNGAP before that. In 2009, it was 7 years later that we just starting getting data from patients. And that wasn't one-off, because it took many more years to start getting more and more patients. And the main reason on that was just the content of human genome, just getting it better and better, not just the draft, and then improving in sequencing analysis. And because of that, we are getting on this position today and finding patients, many more patients. The SynGAP Center was a place that you were doing on a larger scale. A word that you will learn there, it was high throughput. It was high throughput everything. You were sequencing genomes of everything, any organism that you can get. Proteins, which is the counterpart of the genome, is just addressing how many proteins a particular cell or compartment has. We were deriving new technologies. There was a particular project, what it was called the mouse project, which it means that just wipe out every possible gene that they can do from the mouse. So you would get more than 20,000 different lines of mice. This is not very well known. They have several thousands already done, and they are for free, really, for investigators. But it was that kind of thing. So when I moved up from there, I still have that idea of working on my lab at the Silka Neurogenetic Institute in LA from USC. It was this approach, working on a larger scale, not focusing in one molecule. So at that time, still SYNGAP was that one molecule that I have this interest, but among many other molecules at the synapse. So still we were publishing during these years on SynGAP, but with that thing in mind, I say this is just one more component of the synapse. And then I think it was 2017 or 2018 that I met Hans, that Mike, and the SynGAP Research Foundation, and I started building up this connection with them and getting knowing the patients, the kids with mutations in SynGAP. And then to me, this is what I would call the rediscovering SynGAP and starting being back connected to this molecule of the synapse, and still working with this system biology approach, but probably with a focus on SynGAP now it's in the lab. So as I say, it's still the system biology approach, and we still, the Sanger got me imprint on me this thing on not looking at one molecule at a time, but trying to get in within the context of all the molecules that are surrounding your protein of interest in this case. So there was how we describe SynGAP or how we see SynGAP. You have seen this picture many, many times. So SynGAP is what we describe a modular protein. You have different domains that you can see here with these funny names like pH, which is a protein domain that binds other proteins or lipids, a C2 domain that binds calcium and allows regulation of calcium, SynGAP by calcium, a RAS-GAP, which is an enzyme kind of function. So it's a domain that performs a function in the cell. A region of the protein that not many people talks about, which is a domain of an unidentified function, but it's a structure there that nobody knows what it does. That's the name, the domain of unidentified function. So you don't know what it's doing. And at the C terminal, let me see. Oops, because I cannot see. Okay. And at the C terminal, you have a PDZ ligand, which is also a protein domain ligand that allows SynGAP to bind to other proteins. But what I'm showing that is that I like to see the proteins as, or these regions of the protein that we call protein domains as modules of functional modules. I like to define what a molecule can do by the composition of the protein domain. So in this case, you have, what is the function of SynGAP? Well, it's defined by all these collections of protein domain that it has. And why is this important? Because those domains, they might have a particular function, or they might not have a function per se, more like just to associate to other things, which is the case of the pH, the PDZ ligand, and some other regions on the molecule. So I'm not going to describe much what is known about the RAS-GAP domain of the PDZ ligand domain. You may hear many talks on that. Most of the work that has been done on SynGAP, it's mostly associated with the PDZ ligand domain. What is it doing this region of the protein? It's not much known what are the functional consequences of mutations in the RAS-GAP domain or why this really needed at the synapse. So you might be, probably you've seen from other talks, these kind of pictures. This is, you have a different collection of cartoons, a picture of what is the synapse. So you have here a synapse, which is composed by the presynaptic part, which is released neurotransmitter, and the postsynaptic site. Oh, no, it's not working. What you have here, the same thing, but on an electron microscope picture of the presynaptic and postsynaptic site. These are synaptic vesicle there, and here is the postsynaptic site with this electrodense region. This dark region there is what we call the postsynaptic density, or PSD. You have a high amount of SynGAP at this region, which as you probably know, and it's one of the most abundant proteins there. So good thing with this, well, no, good thing why we know so much about the PSD and the molecular composition of the PSD is because we can isolate by biochemical fractionation. We can get a synapse and isolate that part and interrogate and ask what kind of molecules are there, what amount of molecules are there, and what I was going to be very specific. But what kind of modification those proteins have? We analyze and we do different things, like you can analyze behavior in an animal model, do different patterns of electrical activity, and analyze what's happening with those molecules. So in that region, I have said that SYNGAP is one of the most abundant proteins. You have a collection of around 2,000 different proteins. So SYNGAP is one more of these 2,000. It's just one of these 2,000 different proteins. It's one of the most abundant, but you have tons of other molecules there. So one question is that we'll be trying to address through the years, that what are the molecules that SYNGAP binds to? And why that's important for us? Because molecules that are associated together, that just physically interact. So you have one molecule that they just bind to the other. Molecules that have that particular the molecules that are associated one to another are usually associated with the same disorders, and the mutations in one or the other of these molecules are also associated with the same kind of phenotypes. If you say that one molecule, a mutation in one molecule changes the electrical activity of the synapse, well, a molecule that is associated with the other, a mutation in that one also might change the electrical properties of the synapse. Because of that, they usually are connected with the same disorder. So that's something that I've been working for many years, trying to address, not just for SYNGAP, but for many of these molecules at the synapse. Is like, which ones are associated to which others? And we were doing this with this kind of equipment, which is called a mass spectrometer, and we were doing the method is mass spectrometry. So basically, what in this case, what we are looking at proteins, what the mass spectrometer will tell you is that you just put anything there, any preparation there, and it's going to tell you what proteins are there. It's basically working like that. So for example, if we know that we can isolate here the PSD, we do a preparation of a PSD. So when it says isolate synapses, PSD from neurons, we just put that preparation of the mass spec, and it's telling you what proteins are there. So that's why we know that you have around more than 2,000 different proteins at the PSD. You can be more specific. For example, you can get a tool, which is in most of the cases an antibody. And this is, for example, SYNGAP. You get an antibody against SYNGAP. You just capture SYNGAP from the synapse and just put that to the mass spec. And what it's going to tell you is, obviously, you are going to find SYNGAP there, and you are going to find if there are other proteins associated to SYNGAP, you are going to identify what proteins are associated with SYNGAP in the preparation. So, and that's what we call, for example, a SYNGAP interactome, which it means what are all the molecules that SYNGAP can bind at the synapse. So, and we have been doing these things for many years. You have lots of variants that you can test. You can test this and mutants in animal models, like having mutation in SYNGAP or for any component of the PSD. You can do changes in electrical activity and measure how the associations of different components change it. So there are different variants of working with that, and we'll be doing it through the years. These are the kind of molecules that SYNGAP binds to. And you will see it's a collection of these things, that triangles, rectangles, and things. But what it means is that these are molecules that are fairly similar to SYNGAP. There are molecules that also bind to tons of other molecules. So what it means there is that SYNGAP is one of these components that are associated in this region of the synapse. And there are many molecules that share their characteristics and bind to many others. It's part of an intricate, what we call protein-protein interaction network. So there are lots of proteins that are binding together. And this is the kind of what I wanted to convey to you there, and we'll see in the few slides, is that SYNGAP is not isolated. We are not trying to address what is SYNGAP doing alone at the synapse, but it's within the context of all these molecules. And this is, at least for us, is a very, very important point, because the composition of these synapses is not the same always. We are used to, and most of our work has been in adult mouse models, and we are used to and know a lot. I have published more than 20 years of work on SYNGAP and what is it doing at this place. But SYNGAP is doing what is it doing within this molecular context, because it's associated to these molecules at this particular time and place for the cell. So this is not much interesting to see for you. It's just to have an idea of the amount of different proteins that we are getting associated with SYNGAP. So what it means there is that SYNGAP has the possibility to regulate the activity of all these proteins. You can see some of them as being targets. Some of them, if you know that mutations in SYNGAP, and some of them are enzymes or proteins with particular function, you can try to address what is doing a particular mutation in SYNGAP to the function of other molecules. So that approach is telling you you're doing this just to see, okay, are some of these proteins can be targets? Targets, it means like pharmacological targets. It's something that we can do in a model of SYNGAP, like to trying to recover functions, not just of SYNGAP, but some of these other molecules. So that's just, there are many of these molecules. So if you, what I was saying before, these interactions, these protein interactions from SYNGAP change through development. So the molecular context, it's different at different developmental time points. SYNGAP doesn't bind to the same things in an embryonic brain in a mouse to an adult synapse. You have different partners. What you can see here is that what we define in protein interaction network. So here is SYNGAP. Each dot that you will see here around is a protein that binds to SYNGAP. You can do the same with other components on the synapse and trying to visualize what things are in common with other molecules. Why we would like to see what things are in common with other molecules, because all of these molecules that are here, like you see this name here, DGAP1, SHANK3, DLG4, are what we can consider disease molecules. Mutations in these molecules also cause a number of disorders. If you can see here in color code, all these colors means like these mutations in these genes get you a disease. Why is this important? Because it's understandable, and I don't know how focused always you will need to be focused in mutations in SYNGAP. But there are lots and lots of things in common with mutations in these things in terms of signaling mechanisms, in terms of phenotypes that you get in mouse models, and in phenotypes that you can observe in patients. They are part of a protein interaction network that regulates synaptic function at different developmental stages. So the other thing, what I was saying there is that this is a network of proteins, a protein-protein interaction network, and why we would like to see to build up these networks and not just focus on SYNGAP, even if the other molecules are involved in similar disorders. Because networks in general work, any kind of network, biological networks, social networks, in this case, what we have here is an airport network, works in a similar way. You have molecules that are connected to other molecules, and molecules that are not have many connections, and molecules that have not many connections. The molecules that have many connections, we call them hubs. And for example, if you get here is LAX, and it's a big circle there, it means like has a lot of connections. If you get some of these airports in South America, they don't have many connections. And what it means is when you disrupt in a network a component that has a lot of connections, you have the probability to disrupt the whole network. So if you have a slight delay in LAX, you can see that you are going to disrupt flights all across America, Europe, and the whole world, really. If you have a delay in one of these airports in South, or say Buenos Aires here, well, nobody will care much. You are not going to have delays all over the place. And this is the thing with the molecule that I showed you before. All these color codes, all these things that you have there, these are what we call hubs. And SYNGAP is a hub in the synaptic network. Mutations in SYNGAP, because it has a lot of different connections. It's not the only one, you have many hubs. I told you that you have more than 2,000 proteins. So even if there were many things there in that picture, it's a very constrained and focused network. It was, I can understand that you will see there and you see that tons, but there are no tons. It's a very defined network. So SYNGAP is one of these hubs, and mutations in SYNGAP have a high probability to just disrupt the whole protein-protein interaction network, as some of the others that I showed you in that picture. So that's why it's our interest to try to build up this network, because since years ago, we didn't have this information to try to build it up ourselves. And now we have a pretty good idea what is the protein-protein interaction of the synapse, and allow us to do different manipulations now. Again, this is in adult mouse models. And I keep repeating that because I'm going to, if I have time, because I keep talking, is to describe about human models. So these are some names that I say. For example, these are all hubs in those networks. NMDA receptor, which is a subtype of glutamate receptor, and probably you heard about the foundation cure green. This is another hub of the network. This other molecule, which is CNKSR2, DHE3, 4, DGAP1, SHANK. These are all molecules that are associated with SYNGAP in the interaction network. When you analyze mouse model, all of these molecules are going to have deficit in learning, memory, synaptic plasticity, different dysregulation of kinases, even ERK2, that probably you heard that you have dysregulation of ERK2 activity in SYNGAP. You can do any animal model on this. You are going to see changes in all of these things, and you can keep reading and reading papers and finding common things. But in humans also, the mutation, you have mutations in humans associated with these disorders, and you will find phenotypes that you will know, and you will notice also from SYNGAP patients with many of these causes: intellectual disability, epilepsy, psychiatric disorder, sensory system dysregulation, and they are all developmental disorder molecules. So all of this introduction on being SYNGAP, it was just trying for you to get the message that SYNGAP belongs to a large group of synaptic proteins, and I say synaptic between brackets because we know that has functions that are not just synaptic. And mutations in this set of proteins are associated to a number of neurodevelopmental disorders, and they have, some of them have many common phenotypes. And for you, it's what is, so you don't have many parents associations for many of these, at least as the ones that I know, which is the cure green. But I can see that even if that there will be a common ground in mechanisms and trying to join together and reach some other patients with mutations in these things. Sometimes you might see that a phenotype that you might have in one kid might be more similar to mutations in one of these than to another SYNGAPian, for example. You have this kind of variation. They are part of a global network, and mutations in these components are really associated. So just to put together SYNGAP within the context of all these molecules at the synapse. So what about everything that I say at the moment? It was our knowledge in mouse models through the years. So there were a couple of things in the last, I would say, 5 years that also it just turned my lab really more to, still we're working with animal models, with mouse models, but really focus, give us a focus in human-derived models. And this is the capacity of generate, to reprogram cells and generate neurons from a patient sample, that it can be a skin or blood. We usually work with blood samples. And the other is CRISPR method from genetic engineering, that you can these days start just doing mutations, correcting mutations on a larger scale. These two methods just allow us out now, at least to incorporate now mouse models. So we are doing, in general, we have 2 different mouse models, and I'm going to give some info on one of them. We can do, and I'm going to show you some other picture from this, but we can do what we would call a homogeneous cell line model, like you do in neurons in a dish, or we can try to do an organoid type of model, which is a model that tries to capture the cellular complexity of a developing brain. So I'm not going to describe what we are doing with the organoid models. So for this, I'm working with Jorge Quadrato here at USC, and now a new postdoc in her lab, which is Marcella Virtele, which she just arrived from Sweden and is in quarantine now, but hopefully she can start working with us. And for this project in organoids, which we are trying to address SYNGAP function in brain organoids with different scale resolutions, going with single cells, multiple cell types, the electrophysiology levels, and different methods. So we have a SRF funding for this, which is a project that is ongoing, but I'm not going to describe much on what we can do with that. But we're going to define what are we doing with this, just to keep the trace with the neuronal phenotype that we already know. So usually when this is the two ways that we would like to see how to use these models. From the pharmacological point of view, we will try to find, trying to what we would call improving or managing phenotypes, like you would say the epilepsy, eating disorders, sleep behavior, or you can go, and that would be just with particular targets and compounds. And the other thing, you can go and use your model with stem cells and gene editing, mostly for, you don't need to know much about what is it doing or cellular composition, but you would say, okay, we were trying to recover and improve SynGAP protein levels or the protein function. For those for both approaches, you have at least 4 different things that we are focusing. One is to get a target. In this case, it would be a particular molecule, as was saying, within the synapse, for example, or in this case, obviously your target is SynGAP. You will have a compound for that target, or the gene editing. You will need a delivery mechanism, and you will need a proper model. So the thing that I'm going to describe is what it means, this proper model. How are we using the model? Obviously, all these things are important, and I'm just referring to what is our model. So why we're using human models. So human models at least have to me, 2 main characteristics that's why I like them. Is you're working with the proper gene, with the human gene, with all the regulatory elements, and this is absolutely essential for all the gene editing approach. And also you are working with the genetic background of that particular patient. So it might allow you to answer questions like, okay, the phenotypes that we are getting or all with these particular patients, what is the component of the SynGAP mutation and what is the component of the genetic background of that patient? That could be higher, lower, or maybe not important at all, but it is something that I think that we will need to answer in the next few years, or at least as soon as possible. What is the component of the genetic background for the phenotypes that we are observing in the patient, within different patients? And as I said, with the human models, you have a homogeneous model, which is a single cell type. We can do these days a glutamatergic neuron. You can do a GABAergic neuron, which is inhibitory neuron, serotoninergic neuron. And that is something that we like to do because we have a homogeneous cell population that we can answer a particular question, what is doing SynGAP in that cell, or the heterogeneous model, which is in brain organoids. Important to know, this is the first thing that the first time I'm saying this, I'm going to repeat it many times from here to the end of the talk. All these human models are immature models, are fetal-like models, and they are poorly characterized. What it means with this is the synapses that you are getting from these neurons, it could be in a brain organoid or in single cell types, are fetal-like. These are not the same synapses that we were being used to work through all these years of SynGAP biology. What they are resembling is not this mature synapse at the mouse, it's just a fetal-like synapse in a human, and I will show some data on that. So for both these models, you might have 2 approaches, and you can, this is a thing that this is modeling of cell therapy approach. So for example, you have a SynGAP1 patient, usually we work with a blood sample, you reprogram those cells, you then induce pluripotent stem cells from that sample, and then you would like to differentiate it to neurons, a particular cell type. And what you are doing in general in this case is just trying to have a model in a disease in a dish, in a single cell-like single cellular non-homogeneous cell types. Or there are also approaches these days that you are trying to repair those cells and get it back to the patient. But this is not something that we are much interested for SynGAP. SynGAP is a molecule that is just in the whole brain. Most of the cells express SynGAPs. But there are approaches that I will go to at the end of the talk that might be of interest for this case, not just to get back healthy neurons, but it might be something to consider, or at least that we would like to explore is neuronal progenitors of particular cells that might be of use. And this is something that we are starting to address, but that will be discussed at the end. So thanks for, with many of the patients, I don't know how many parents are, might be discussing how many parents gave me, that might be hearing now, gave me samples for studying SYNGAP function, but we have a good number of mutations of SYNGAP. These are just these balls there is where are the mutations located in many of our samples. I'm going to describe some data with a mutation that is a truncation mutation here at the RAS-CAP domain and a different mutation on splicing site more close to the C termini. So this is some data. Let me see. So when you get your sample, what you always get with, well, and you generate the neurons, one thing that you always do is what you correct the mutation. So what it means is that you just put back the normal base, but whatever is the mutation, you just correct it back to normal, and that's your control. So you are controlling there for the particular genetic background of the patient, and you need to do that for every single sample that you do. In this case here is what is called a Western blot. What you see, these things there are SYNGAP, all on this line. And what it means, C is the corrected patient. So you see, what you see in a Western blot is what you measure here is an intensity. It's not a good way to measure anything. We do everything for mass spectrometry. This is just for illustration there. I hate these kind of representations, but it's useful for talks, and also reviewers like them, and they used to complain of silly things, what is the size of this everywhere, because it looks like if you see it, it's real and not in something like a mass spectrometer. But well, this is how in science works. Is a mini rant on that. But well, this means here is a corrected patient. This is a knockout. This is a patient that doesn't have SYNGAP at all. So it's a control for us. And this is the patient. This is corrected, knockout, control. You can see that. And this is wrong because this is the patient and this is the corrected. So this is the patient, you don't see much SYNGAP, and in the corrected, you have more. Again, this is the patient, this is the corrected in another clone, and it has more. This is the patient, nothing, corrected, you have more. So you will say that in this case, that mutation decreases the amount of SYNGAP from that patient. To what extent? Well, you will say you can do a quantitation on this, and you can say that is around 50%, which you would call a haplosufficient model. And I would say something of this about the end of the talk. This is not a good way to do it. And I was just showing by Western blotted, but we will need to get better. And this is we're working with this to generate a reliable model to really properly quantitate for each mutation in patients how much SYNGAP is in there. And there might be the cases that you will say, yes, it's a 50%, roughly 50% of something, but there might be that some patient have a 50%, another patient has a 65%, and that makes a huge difference in the phenotype. I'm not saying that that's the case. I'm saying that we don't know, and this is a very big concern that I have at the moment. So we can derive neurons. You have different pictures of neurons that we do. These are the human neurons from patients with mutation in SYNGAP. So you're not going to have the eye of what it means to be an immature neuron within mature spines, but you need to believe me that when you see spines like this, like you see like not much a shape, like a hair, if you want, or something that's protruding from this green line, which is what we call a dendrite. These are immature spines. You would like to see spines more like this. You see like a kind of mushroom thing, and you would like to see tons of them. When we were working with mice, you will have tons, just complete, the dendrite completely full of these things. These are these neurons are behaving like an immature cell, even if this is and the difference between this and this is because these have been kept in culture for 3 weeks, and in this case we will be kept in culture for 8 weeks. And you will say, okay, you can keep them in culture for 6 months, you get it better. Well, you're not going to get much better. We never got it for 6 months, but you're not going to get it much better, because these are human cells and they follow the developmental profile of a human cell. So even if you culture for months and months, you're not going to get to that. And also it's really impractical for any purposes, just getting an experiment to run for years. Oh, again, if you see where it will be SYNGAP around there, is SYNGAP will be, I don't know if you're going to see it, but SYNGAP is going to be at the tip of that spine. That red thing there is not SYNGAP, it's PSD95, which binds to SYNGAP. So SYNGAP is generally in that region. So one thing with this is that when we are working with synapses, and if you see this, you will straight away think that it's very, very difficult to work with SYNGAP in these cell models because you don't have many spines. You don't have a lot of SYNGAP in these cells as in a mature synapse. So that's what we need to get better and better in trying to characterize the cells and trying to develop models that allows you to work with SYNGAP in these cells. So bottom line with this is it's not the same cell type, it's not the cellular context is different, and it's really difficult to work with SYNGAP in this condition. These are, for example, with one of the samples from a patient, a corrected cell line that you see firing of those neurons that we can record in multi-electrode arrays. And this is what you say control or a corrected cell line, and this is the mutation. Just if you look at this, you will hopefully you see that the one at the bottom with the mutation, you see more firing. So at least these things are burst. Is a measure of the network activity of your culture. So you will see that it's more, and you will say here is you have some trace recording here. You will see that you have more burst in the patient, which is more like you will say is firing more in this case. So I know how I'm doing with in time. It's not good, is it?
No, I think I'm hearing the people are enjoying this a lot. So please keep going.
Because these are just pictures on showing proteins that are present at those neurons that I said that are immature. So the first thing to me was in characterizing these cells, it was, okay, what things can we do in a cell that is in a neuron that is a fetal-like neuron? And to me was the first question is, okay, we had SYNGAP there. How much SYNGAP in relationship to other molecules? Because we knew that it was one of the most abundant proteins in the mature rodent synapse. Do we have all the other components, these components that we say that are associated with disease and all this protein-protein interaction that I show you, are there? And first question to me was even if those cells had PSDs or no, which was this thing that I showed you before in an adult mouse, that is electrodense region. Well, so I spent hours on an electron microscope myself trying to see on this. And yes, I got pictures, and here you see this electrodense here and in the red part there. But what I would say is that there's not plenty. I spent hours and hours just chasing PSDs. So, which is obviously obvious because you don't have so many spines in this cell. So it's difficult to get. But next thing is, can we characterize those PSDs, even if you don't have many synapses? So these are Western blots. And these are all names of molecules that we thought that we were interested to see if they were there or not. So this is, if you say, when you say sol, it means like that's not the synapse. It's, for example, soluble. What it means is like these molecules should be around here or there, but not here. Whatever is PSD, it means things that we are isolating from here. And this is like something like you would say a control. So if you say, this is a subunit of an NMDA receptor, we look at the PSD, yes, we can see it. In the other region, not much. The same with another subunit of NMDA receptor. This is a different type of glutamate receptor. It's in both as expected. I was expected to have it in both. This is a protein that it should be localized here at the presynaptic site. And you see that we don't have much or anything at the at this end, and it's there, so which is good. AMPA receptor, which probably you hear a lot. I don't know if you hear a lot, but you should have hear a lot about AMPA receptors in some of the talks. But again, we can find it there, other molecules, and we were pretty happy. This doesn't mean much. This means that the molecules are there. And next thing, I was looking on a human fetal brain, and we found similar patterns. So what it's telling you is not much. So what you will say is that in a human fetal brain, you have this molecule that we have been studying as well in a mature synapse. The amounts are not the same. It's difficult to work with, but to me, at least, this is telling you that you can still answer questions related to SYNGAP function at the PSD in this cellular model. You will need to be careful, and I'm not going to go into many details on this because the composition is not exactly the same of the molecule, but it still is something that can be used. So you will see many in different kits that you've seen. You are obviously you don't see the same phenotypes. Different mutations get you also in a dish different phenotypes. For example, this is electrical activity, and you say here is the patient with a mutation 1. You see this pattern of electrical activity, which it means that is higher comparing with the control. You have in this mutation here, you see the pattern of electrical activity is completely the opposite. It's lower. And that's what I was saying. You will need, if you want to get the proper characterization of genotype, which is the mutation, and phenotype, which is your observation, you will need to know very well what the mutation is doing to the protein. How much protein, if you have a truncation, if here is produces some partial, it's just a partial protein that you get. If it is a complete wipeout, then you have haplosufficient model. This is something that we need to get better to know. You can analyze our numbers of spine, morphology of the spine. You see, for example, in the first case, the phenotype that was having a larger electrical activity. You see that, well, you're not going to see much here, but you will need to believe me that you have more spines in this case with this mutation that in the corrected. That you look at here, you have what we call a spine intensity. So it means like in the patient, you have more spines, and also they are bigger. You have in the second case, you don't have any difference in spine density and also volume, which is not shown there. So you have different mutations as you expect or not, but you have different mutations. They are producing different phenotypes, and the clinical phenotypes of the patients also, it's different. Next thing that we would dig with this, because for us it was important, is okay, it's all these things, as I was saying, that you don't have many spines, and the amount of material in each one of them is not as much as you will expect from a mature synapse. It's like, can we do biochemistry? Which is biochemistry means that work with the proteins in the same way that we were working in a mouse model. So we were trying to do the same things that we always did with the mouse model, isolate synapses, isolate the PSD, and start interrogating what is in there by mass spectrometry. So we were doing that, and I'm going to show some pictures, and don't pay much attention because there are lots of numbers and things. So the only thing is for that is that we can monitor what's going on in the synapses from derived from patients and with different patients. And we can see differences between one or the other. What it was, I was saying at the beginning, a larger scale with what I would like. So for example, we can prepare these PSDs from these neurons. We can correct, for each one of them, you have different clones. I'm not going to technical, but things are not very reproducible when you work with this. So you will need to have tons of control. So these experiments were working really, really nice because they were really, really reproducible between different clones. And you can start quantitating changes in the total number of protein or changes in particular, what we call, this is going to be awful, post-translational modifications, which is protein phosphorylation, for example. These are things that you produce a change, which it means increase or decreasing phosphorylation in a protein, and that produces a change. What it means is the cell needs to communicate what is going on from outside to the inside. And how does it do it? You have a molecule that senses something and then transduces what it senses to the inside of the cell. Lots of these things happens by what we call protein phosphorylation. So it's getting you an idea what's going on and how you respond to that. So by monitoring changes in protein phosphorylation, what you try to address is that what's going wrong in all the signaling mechanism. So, okay, you have a mutation in SynGAP, but you want to know what is wrong within all the cellular mechanism, the signaling mechanism of the cells. So when you do it at a larger scale, you have a pretty good picture on that. And we got like more than 1,000 phosphorylation sites and in these samples. And we can monitor changes in phosphorylation in one way associated with one mutation or another. For example, in this mutation, we got more than 100 phosphorylation sites that being changed, and in this one, we were more than 300. You have some going up, some going down, and then you start to building up what is in common or not, how it might affect the protein interaction network. So we start saying, okay, which molecules are affected, which molecules are not affected. We're trying to build up the same thing that we will already know from the mouse model with the patients, really. That's the bottom line. The difference is like, okay, we have now real mutations that happen to real people and within the context of a human cell with a particular genetic background. So what I would say with this is that even if you have all these color codes and things that I think we're pretty happy that we have a robust cellular model to monitor SynGAP function in these cells. It's working really well. And it's a lot of work, really, because you can say what things that are not abundant means like you need to culture tons of tissues, and it's limited anyway. Even if you culture tons of tissues, if you don't have many synapses, you increase more background than the number of synapses. But it works well. So we're pretty happy. It's not the same thing, as I say, and I will keep repeating, it's a developmental synapse, but we have a model that we're pretty happy with, that we can monitor changes in SynGAP function. The other thing is like where everything that I was saying is an excitatory neuron. What it means an excitatory neuron that you see here is, I don't know why is this block there, but well, SynGAP used to be in synapses around the dendrites around here. And everything that I describe is what we know in excitatory neuron. What is a mean excitatory neuron is like what it releases here from that terminal is an excitatory amino acid, which in general, in this case, is glutamate. That's why this is an excitatory neuron. You don't have just, SynGAP is not just expressing excitatory neuron. You have lots of different excitatory neurons, different types of neuron, depending on what is releasing those vesicles. If it is glutamate, in this case, is the neuron that I described, an excitatory neuron. If it is GABA, it's an inhibitory neuron. What is going to do that neurotransmitter on the neuron that it makes a synapse is just inhibit that activity. It's a complete different thing. So even on all this, if you have dopamine, dopaminergic neuron, it releases dopamine, serotoninergic neuron releases serotonin. So most of what we know, it's the description is this type of neuron. SynGAP is expressed in many other neurons, and we don't know what is it doing. We don't know what is it binding. And really, we don't know if it is really, what way can that help us to understand or find new therapies for mutations in SynGAP? But I was just a little data on that. So good thing with these human models now is that they are pretty homogeneous. When I say pretty homogeneous is that, okay, we have protocols from a blood sample to do an excitatory neuron. And most of those neurons in the dish are excitatory neuron. And this seems obvious, or maybe it's not so important, but when you're used to work with a mouse brain, getting this level of homogeneity and just to address particular question is very difficult because you cannot get just an excitatory neuron from the mouse brain. You get a mix of things. So getting just one particular cell type on your dish, for us, it's really important. In that, at least for answer a specific question. So we did the same, and we just, as I showed you for excitatory neurons with GABAergic neurons. So this is an inhibitory neurons. And you can see this big blob there is that SynGAP in excitatory neurons. to our surprise, there was quite abundant. This is a very relative thing. What is abundant or not? I could say that it's comparable to what we found in the excitatory neuron. So obviously we can immunoisolate here SYNGAP, which is what is shown there. We can get it to the mass spec, see what is it binding there, and start to address questions of what is it doing at the inhibitory neuron. And when you start playing with different neuronal types, you can start doing different things. For example, if this is an excitatory neuron, that would I show you the electrical activity here, which is a multi-electrode array. These are electrodes, all these things there. And you see these kind of white things are neurons. So that you record the electrical activity there. That's what we showed before with the excitatory neuron. You can do the same with the inhibitory neuron now. And then you can start playing with things like, for example, culturing an excitatory neuron and an inhibitory neuron and measure electrical activity. You can measure activity of both mutations in excitatory and inhibitory. You can have in your dish a mutation in SYNGAP only in the excitatory, but you can have a dish with mutations in SYNGAP only in the inhibitory neurons. We are doing the same thing also with dopaminergic and serotoninergic cells. Important thing with this is, as I say, is that if we can address some of this function and something that it might be interesting in terms of a question that I would say, is it important to at least restore SYNGAP function in inhibitory neurons? Because there are protocols and they are being just, at this moment, there are being clinical trials on patients being treated with neuronal, it will be neuronal progenitor cell from inhibitory neurons. What you get in your brain is a cell that is going to become an inhibitory neurons. And that's being treated mostly, the idea is we will have some work on epilepsy. But you can think about is that if really SYNGAP has a function and is important in inhibitory neurons, that might be something that might be worth to explore what it might be and the consequences of what might be the improvement in restoring SYNGAP activity, at least just for inhibitory neurons. That's why we are interested in this. Just I think to finish, genetic background effect. I think it's something that we like to, and we are already doing this, is just to try to address what is the effect of the background of the mutation. What we are doing with this, for example, you have here a mutation, which is this in purple circle there. And usually what you do is you correct it, and you turn it to green. You have a different patient with a mutation here in blue, and you correct it. What we are doing now, when we're trying to address is that what is it doing the mutation from, for example, the patient 1 within the genetic patient 2 with the genetic background of patient 2. So what you have here is that you correct one patient, and then you introduce the mutation of a different patient in the corrected background of the first. It looks it's difficult, but what you are doing is as you just correct it, you get a normal cell line, and then you get the mutation of the other. In theory, and then in that case, you say, okay. We're really interested in this, in trying to address is that, for example, haplosufficient model would get you the same phenotype in one background or the other, and different types of mutation. This is something that we don't know. And then it's obviously you will try to get the different characterization that we are doing. So to finish. So I think that this is really important and something that we are pushing forward and trying to determine the effect of a mutation in SYNGAP protein. It's what is really doing when you get from your genetic counselor, it says mutation in SYNGAP and it's predicted truncation or something that's predicted. And we don't have a good method now to tell what is really going on, what is really happening to the protein levels, or at least to the structure of the protein. We are analyzing different cell types, and we will need to have this better characterization of cellular model, although we are kind of different cell types. And what we are getting, yes, we expect to get this method pretty soon. We are trying to do it by mass spec. We have at least a cellular model to address this. We are also getting those different cell types to try to address alternative targets and pharmacological approaches for cell-based therapies. And this characterization of the models that I say, I don't know if that was really clear, but one thing that is really important, if you are going to move forward in gene editing approaches, these human models are the proper way to address it. You are having the background of the patient, you are leading with the gene, with the human gene of SYNGAP with all regulatory elements. So it's what we're likely to try with these models now. This is the people that have been working in the lab, people doing mass spectrometry, collaborators in different UCs, and Georgia and I'll say Marcella that we are carried forward with the help of the SRF, the organoid model that hopefully, probably she, Marcella, or I, anyone can talk pretty soon next year when we don't have so many restrictions with COVID and the things that we can start going back and work at full force at the lab. So thank you. And I don't know if you have a question. I think that we are getting questions afterward.
Yeah. Thank you so much, Marcelo. I think you can take down your slides. I've enjoyed exchanging messages with a few families during your talk, and all the scientists and people with medical training have enjoyed all the science, and the non-scientists are also enjoying knowing that there's so much good work going on. I want to just start by saying how grateful SRF is that we are in touch with you and that we're working with you, and I'm glad we have this webinar just so I can show families the breadth of your work and the depth of your knowledge, so they can understand why we're working to get you a postdoc in your lab. But as you know, because we've had some conversations before, the families ultimately are thrilled with the science, but want to understand really simply how research will affect treatments for their kids. And as a pharmacologist, you think a lot about treatments. So can you just in as simplest terms explain to parents why your work in human models, right? So you made a very good point earlier. A lot of people would do work in mouse models of one kind of neuron, but now you're doing human models, and you're putting, one kid's mutation in another kid's background so as to see what we can learn. What can you just, in simple language, answer why is this going to help us as we start thinking about therapies?
Yeah, I would think one of the things, you need a model to at least, you need to characterize well your model to know what you are looking for. You're saying that you are trying to correct SYNGAP.
Right.
What does it mean? What are you going to look for? Yes, we are focusing getting model. It's in, because what you get is that there are people from lots of different places trying to do this with lots of different disorders. It's just to correct a mutation, really, with lots of different things. An important thing, and I have this talk with people with pharma companies and people working, they don't have a clue on what to look for.
Right.
And what are you going to get? You're trying to model your classic mature neuron and trying to see what is SYNGAP doing there when you don't have many of these molecules. Are you expected to almost have very, very little amount of SYNGAP? How are you going to monitor changes in, you edit SYNGAP and you say, okay, you have a haploinsufficient, your gene is decreasing. To what level? To what level you need to increase it to start seeing something? These are, if you want, in a way, if secondary things, if you wait, you will say, okay, first I want a reliable method to edit it. We are working on these things as well, but this is not what I presented. What I'm saying is that something more general that needs to be built up is that the proper model system to test that whatever you are doing, it works. I don't see that we have this at the moment. And because the model, I would say you need to do it in a human model, but you need to understand the model that you have. And maybe you don't need excitatory neuron. Maybe you can start working, we are thinking, maybe we are doing in the inhibitory neurons. If we see that in this particular cell, we have more SYNGAP or it's more easy to work with. So these are things that, that we're working with, and it's working fine, but it's not easy. So it needs all this characterization that we were doing, and I think that is something important that needs to be done.
Great. Okay, I'm going to start reading you some questions that have come through the Q&A, and I would encourage the families and the scientists to put their questions there, please. And with apology to the scientists, I'm going to take parent questions first, but I will try to get through all of them. So one question from JR, when you have a fast assay for protein levels for each mutation, will that require blood donations, or will you recreate the genetic variant in your own cell line?
Yeah, when I say fast, it's not like we have a blood test and we say what is happening. It's fast for our standards. What I would say is that, well, you will need to go through these processes. You will need to get your blood sample, get your cell line, culture that cell line, and then when you have an amount of SynGAP, yes, measure in that case will be fast. After you get your neurons, and then you go, it will be by mass spec and knowing what to look for and address. These are things that they need to be like that. There's no way around unless you want to work with mRNA, which I want.
Okay, thank you. Next question. JR is interested in the 2 SynGAP haploinsufficiency models of different ages for the neurons. I think, this is her talking, I think you have a variety of observations in both and that they are different. Is that right?
I cannot, for one of the ones that I showed, for one of them, I can see, and that's what I cannot tell that one of the models is happily sufficient. I cannot tell. I still don't know. That's what I mean. We need a method. It's simple. If you do that thing that you see in a Western blot, you do some samples and you do some stains there and you say there's more or less, but that cannot tell you if one model is 50% and the other is 65%. I cannot tell that. I know that the patients are different. Clinically, they are different. And in my mind, I cannot think that such differences are coming from both being happily sufficient models, because otherwise I don't know where we're standing, really. If every happily sufficient is completely different, are you going to, I don't know, it might be genetic background. There are different things, but that you will need to check. I would really want to know, yes, what is it really doing in each case, the protein, and then you move forward. That's what I mean. The methods at the moment are not telling you, they're not so precise.
Right. One of the things, and then I'll go to Pavel's question, which is at the bottom of the Q&A if you're reading them. One of the things I say to people is, generally when we're talking about SynGAP researchers, a lot of the work is focused on a certain kind of neuron at a certain kind of age. And as the research that's coming out just in the past couple of years about the different isoforms of SynGAP, that you've been an author on some of those, what you're really digging into, and please tell me if this is right, what you're digging into is the different functions of SynGAP in the different types of neurons and at the different stages of development. And that's work that really hasn't been enough done there yet. Am I being accurate when I say that?
Yeah, there are things that, it's lots of time in science, it's not like we're trying to get to the minute details or knowing what exactly to do everything, because what you need is a solution, and I totally understand that, and I'm looking for that. But there are things that it might look minute to you that it's, at least there are things that I need to know first to then move forward. I said, we don't need to know what exactly each isoform is doing in each cell type to get something working on. But there are things that at least just to characterize the model, knowing what to look for there. It's important that maybe in that case is that should we look for a particular isoform in this model? I don't care. I mean, I might not care if it is for correction. For pharmacological approaches, yeah, you would need to say, okay, or build these networks and say where it might be other targets. For corrections, it's different. I don't care which isoform, but at least I need to know which one to look for.
Okay, thank you. And then I don't know if you're reading them, but Pavel's question, if I understand correctly, you're trying to determine some of the compensating effects of other mutations in the genetic background on the phenotype. Could this unearth other targets for treatment, especially in the case of quote unquote mild patients? Do you have any idea of the relative balance between the type of SynGAP mutation and the possible effects of other changes in genetic background and the resulting phenotype.
Yeah, genetic. We're working on that. We will see, but that's when we're working with that. You're getting really specialized medicine. The issue, you're just dealing with one patient.
Yeah.
And when we say genetic background is super, it's really, really wide. It might be something like our just variants, which means you have mutations that per se are not getting you a different phenotype, but they are influencing, which is really, really tricky to know what is going on. But sometimes, and we are looking into this, there are patients with mutations in components that you're saying, okay, this might be doing something else. Because, yeah, it's not just one SYNGAP mutation. At the end, is you have, yes, you might have mutations in other genes that are doing something more dramatic, if you want. You can still go that background, but those are more easy to tackle. If there are variants through a genome, it's, yes, we are doing assays just to see if that has an effect. But yeah, it takes a lot of time because you need to correct one patient and get the mutation on the other, and that's just two. We are not planning to do this for everyone, but at least that will tell us role of background in define things. We are planning to do that in happily sufficient models and say, okay, they're behaving the same with the different backgrounds, knowing what are the clinical characteristics of the patients.
Got it. I think that question's been answered. And then Sydney is asking the last question. You shared about how the molecules that SYNGAP associates with change depending on the stage of development. Would we expect that mice that have their SYNGAP levels reduced would then have an associations with other molecules that is appropriate for the chronological age in non-mutant mice?
Let me read it.
Yeah, it's at the bottom of the list. It's would you expect that mice that have their SYNGAP levels rescued would then have associations with other molecules? Other
than the normal ones?
Yeah, or other proteins maybe.
Well, lots of these things in the cell, it works by molecules being at the right place and in a particular amount. When you say that the molecule binds to another, it's because the structure of these two molecules that you have a shape and the other binds like this, but that's what we call affinity. When you put a lot of something, affinity doesn't matter much. You just go and it doesn't matter. So I don't know if I'm having a correct interpretation. If you say, when you rescue, you see something, it could be that if you're putting tons of SYNGAP, binds to lots of other things. But I don't know if I'm answering that. And in a mouse model, it's okay. I will expect that if you I don't know, it depends on what is the method that you are using for that rescue. But I will SYNGAP is really about, with all what I'm saying, that yes, you would have a developmental phenotype. I don't know if when you get a late rescue, you will, in a human, you will get what sort of phenotypes are you going to recover? That's not known. And I wouldn't care much about that because you cannot do anything from whatever. If you have a rescue, you just do it. And if it works, it works. Whatever it getting work to work, it works.
Yep. Two more science questions. Shan Wang asked, have you checked the EI balance in your human model? I don't know what EI balance means.
Which one it is?
Third from the bottom, Shan Wang.
No, we still didn't do I suppose the excitatory inhibitory, I guess. No, we still didn't do it.
Okay, cool. And then since you're reading them, let's just go down from there. Janie Reed asked, I'm especially interested in the background differences and how they might affect embryonic brain structure. Are you seeing that now?
We still don't know. We are going to do that. Yeah.
Great.
We already did those swaps.
Yeah.
We still don't have the results. Yeah.
So we see that different SYNGAP patients have different reactions to different anti-epilepsy drugs. Is this something that can be investigated using human cell models? Ooh.
Yeah. Yeah.
Okay, cool.
Yeah.
And then,
well, we don't know it. that's something that you can do. Another correlation.
Yeah.
But I would say those models are suitable for that. Other option is going with rodents. Well, you're not doing, you cannot get background, the proper chain. Yeah.
Okay, then there's this, and then there's these three questions from Anonymous. One of them is asking about what SynGAP isoforms are. Another one is asking about, can organoids feel? I haven't thought about that one before. And then the last one is, how can mutations, of which all are expected to reduce SynGAP protein, result in opposite phenotype in cultured human neurons? I'll let you.
Well, that's what I was saying, that we will need to reduce SynGAP protein. That's knowing what is really doing the mutation. If you say that's perfectly understandable thing to say that, okay, you have haploinsufficient that decrease 50% of SynGAP in this patient, and in this one, how are they different? And you will say in cultured human neurons, you might say, oh, that's an artifact, but the patient had different, that's why I have different clinical phenotypes. Mm-hmm. So it's not just that you are seeing that there. But that's what I was saying, is that we will need to be sure of what the mutation is doing to start to answering that question. And if that's the case, you can start being, the next question is just start looking at the background.
Okay, got it. Well, with no further questions, I want to just thank you so much, Dr. Coba, for taking the time to let us see how your brain works. We're excited to keep working with you, and we look forward to seeing what you come up with. Marta, do you want to?
I just want to say thank you. This was great. I hope all the families can understand the work we are trying to do with you, because it's very exciting. I think it's going to help us a lot. And we really hope everybody, got a good course of what you are doing, because it's great. And thank you, Marcelo. Thank you.
Thank you.
Yeah. And I just want to end with that said, we due to just the scheduling and changes in schedules, we do have another webinar tomorrow. So if this didn't make your brain hurt enough, you can come back tomorrow. And I just want to share this. SynGAP.fun Sanders. This is Dr. Sanders from UCSF, who will be talking about the developing human cortex. And since I've got slides up, I'll just go through them. We also have the SynGAP roundtable, scientific roundtable, with a lot of remarkable scientists, including Dr. Coba. So we told Dr. Coba this was for parents and he should keep it simple. So I cannot wait to see and he can talk science at the roundtable. So I cannot wait to see what he presents at the roundtable. But Mary Kennedy, who was one of the earlier scientists in SynGAP and is still busy at it, Caltech, Dr. Heller from Penn, Rumbaugh from Scripps, Dr. Coba's on the screen, Ellie talking about our natural history study, Dr. Smith-Hicks from Hopkins, Al George, who I think is on, glad to see him, and of course, our very own Hans Schleck talking about SRF. So syngap.fun/RT2, please come. We do have a natural history study. You can join right here. If you're a parent who's not on it, syngap.fun/citizen, 2 eyes. We're selling stuff. Everybody loves these sweaters, syngap.fun/bekind, if you can still order in time for Christmas. And then just a reminder, this is tomorrow, don't miss it. And if you have questions or comments or feedback and you want to talk to me, syngap.fun/Mike. Thank you all very much. And special thanks to Dr. Coba, and we look forward to working with you for years to come. With that, I think we're done.
Thank you.
Bye, everyone.
Bye.